Welcome back to this next video in which we are discussing influenza. So we've now discussed the background knowledge that I wanted you to know with regards to the different types of epithelial linings that line the different portions of the respiratory tract. And I just want to summarize this quickly because it's really crucial that we understand this. So I just want to give you quickly the take-home message that we learned in the previous videos. So we learnt about these different types of epithelial linings. We started with this respiratory epithelium, which lines a lot of the respiratory tract. And remember, this lines the nasal cavity, the top portion of the nasopharynx. It does not line the bottom portion of the nasopharynx. It does not line the oropharynx. It does not line the oral cavity. It does not line the laryngopharynx. It does not line the top portion of the larynx. And it does not line the esophagus. It does, however, line the bottom portion of the larynx below the glottis, and then it lines the trachea and the bronchi. And this respiratory epithelium is susceptible to influenza viral infection. So the ciliated columnar epithelial cells of this respiratory epithelium can be infected by influenza viruses. So all of those areas that are lined by this respiratory epithelium, they are susceptible to influenza viral infection. Now, the portions that I missed out in the middle then, the bottom portion of the nasopharynx, the oral cavity here, the oropharynx, the laryngopharynx, the top portion of the larynx and the esophagus, they are lined by the second type of epithelium that we talked about, which is the non-keratinized squamous stratified epithelium. And the big difference is that this epithelium is stratified. It's made up of a huge number of layers of cells, and that this epithelium is continuously being shed. The outermost layer of cells is continuously being shed, and the basal cells are continuously dividing to produce new cells to replace the ones that are being shed. And this type of epithelium is not susceptible to influenza viral infection. This is why you do not get a sore mouth when you have flu. Even though the virus must be, it's very, very close to the oral cavity. Why does it not infect the oral cavity? Because the epithelium is wrong. So it cannot infect these areas. The reason that you get a sore throat in flu is because it can infect the top portion of the nasopharynx and also the bottom portion of the larynx. And when people say they've got a sore throat, they often don't distinguish between whether they've got a sore voice box or a sore pharynx behind. They just mean that they've got soreness in that general region. Excuse me. Those portions of epithelium cannot be infected by the influenza virus. The final type of epithelium that we discussed is the alveolar epithelium, which lines the alveolar air sacs, and that portion of epithelium can be infected by the influenza virus. However, in most people who suffer from flu, thankfully the virus doesn't make its way down that far. So as the viral infection progresses, as we'll discuss later, we'll go into this in detail later, it progresses down the respiratory tract. Usually it starts up in the nasal cavity and it works its way downwards. So it'll go back to the nasopharynx, then it'll skip out this bit, down to the bottom portion of the larynx, down the trachea, into the early bronchi. But thankfully, usually the immune response gets on top of it before it makes its way all the way down to the alveoli, thankfully. And therefore, usually, in most people who are get flu and get better, it doesn't ever get it all the way down to the alveoli, so you do not get pneumonia. However, in some people it does get that far down and you get a horrendous illness called primary influenza viral pneumonia. But usually that doesn't happen. But we do need to be aware that those cells are susceptible to influenza viral infection. So that's the key message from all of this histology that we've been talking about. Which portions of the respiratory tract can be infected by influenza and which cannot. Right, so let's now move on to the bulk of the video. And the first topic that we're going to discuss is the influenza viruses. So let me get some evil colour to put this as the title, because these are, after all, the stars of the show. They're the villains of the show. So, influenza viruses is what we're going to now discuss. So, the first piece of information that I'm going to give you about influenza viruses is not a particularly important piece of information. It's not a particularly exciting piece of information, not particularly interesting. However, it is a piece of information that people expect you to know. People think you're clever if you know this piece of information, and therefore I'm going to give it to you. And so, As I said at the start of the video, there are 3 different species of influenza virus: influenza A virus, influenza B virus, and influenza C virus. Now that is an important piece of information to know. The unimportant piece of information to know is that these viruses are classified into a family that actually includes some other viruses beyond those 3 species of virus. Species of virus that you will never have heard of are also members of this family. So these 3 viruses, these 3 species of influenza virus, they're grouped into a family along with a bunch of really niche viruses that you will never have heard of, and this family is called Orthomyxoviridae. So a big, pretentious name that people expect you to know but which isn't actually important, but I'm going to give you anyway. So Orthomyxoviridae. So of course, this is just the way that someone long ago decided to classify viruses. There is this family of viruses called Orthomyxoviridae family. It contains all the species of influenza viruses and a bunch of other viruses that you will never have heard of, and which I've never heard of, and which I'm not going to give you the names of. So, next piece of information is that there are 3 different species of influenza virus called the influenza A virus, the influenza B virus, and the influenza C virus. Now, these are often shortened down. So, some abbreviations that are quite useful to know are IAV is an abbreviation, of course, for influenza A virus. And by the way, you put the A separate to influenza, so I might actually, just to make sure everyone's completely clear, write out its name in full. So this is short for influenza, that's the I, and then separate from influenza you put the A, and then after that you put the virus, like so. So that's a species of influenza viruses. Like humans are a species, this is a species of influenza virus. Now there are 2 other separate species of influenza viruses that do actually have slightly different structures to the influenza A virus, and we will go over how the structure of these other species of influenza virus are different to the influenza A virus. Now, by the way, influenza A viruses are the worst. They cause the most severe illness compared to the other 2. Okay, so the other 2 are sensibly called influenza B virus, IBV for short, I won't bother writing out its name in full again, and then influenza C virus, ICV for short. Okay, so there are the 3 species of influenza virus, and they're all in this silly family called the Orthomyxoviridae family. Now, I use the analogy of a species of influenza virus to the human species. When you look at all the members of the human species, there are loads of different members, and they're all slightly different. It's exactly the same with influenza A virus. If you were to look at every single influenza A virus that exists on this planet, they would all be slightly different. They are not all identical to one another. They are different. So we often talk about different strains of influenza A virus and different strains of influenza B virus and different strains of influenza C virus. So this is an important concept. So each of these has lots of strains which are different types of influenza virus. They have significant differences from one another. So you have this species called the influenza A virus, and then you have loads of strains of influenza A virus that are all different to one another, and the immune response to one strain of influenza A virus will not be the same necessarily that is needed for a different strain of influenza A virus. So this is why you can get flu multiple different times, because of the different strains of the 3 types of influenza virus. If there weren't different strains, if all of the influenza A viruses and all the influenza B viruses and all the influenza C viruses in the world were identical to one another, then you'd only be able to get flu 3 times, wouldn't you? You'd be able to get influenza A virus flu, influenza B virus flu, and influenza C virus flu, and once you'd had all 3 of them, you'd be completely immune. It unfortunately does not work that way. For each of these species, there are loads of different members of that species. We call these the different strains of the influenza virus, and they need different immune responses to destroy them, and unfortunately that means that you can get flu a huge number of times in your life. Okay, right. So, the next thing that I want to do then is now start actually discussing the structure of these viruses. And the one that we're going to begin with is, of course, influenza A virus. Where else would I begin? So we're going to start by describing the structure of influenza A virus, and then we'll go on to look at the structure of influenza B virus and influenza C virus, and we'll see how they're different from the influenza A virus. So, conveniently, I have already got a nice title there. Influenza A virus, so I can just use that as my title. So we're now going to do the structure of influenza A virus. So all of the influenza viruses, they are all enveloped viruses. So the outer layer is going to be a phospholipid bilayer that they have nicked from the host cell that made them. And they're also RNA viruses. So this is a key piece of information. Their genome, their genetic information, is not encoded in DNA. They do not have DNA inside the virus particle. Instead, they have RNA molecules that is encoding the genetic information. And this RNA is single-stranded RNA. So there are a huge number of different types of viruses. Some of them are DNA viruses, some of them are RNA viruses. When we're talking about the RNA viruses, there are single-stranded RNA viruses and there are double-stranded RNA viruses. And then when we're talking about the single-stranded RNA viruses, there are again two different types. There are the ones where the single-stranded RNA is positive sense, and there are ones where the single-stranded RNA is negative sense. And in influenza viruses, it's actually going to be negative sense. And remember, the meaning of that is that the RNA that the virus has inside of it cannot actually be used to make proteins. It's not the right piece of RNA to make the proteins. You can't translate it into proteins. You have to take the complementary piece of RNA, and then that can be translated into proteins. So positive sense RNA is RNA that can be read and translated and make protein. Negative sense RNA is the complementary strand to that can't be read and translated into proteins. But I'll go over that all again when I actually come to the genome of influenza A virus. So, first thing to draw then is the outer envelope of the virus. So, we'll do it here. So, we'll have the envelope in this blue colour here. So the virus is a sphere, it's a spherical virus. Here is the envelope, so I'll just label this up. So this is the viral envelope here in this bluey colour. Right, now this is a phospholipid bilayer, so it consists of the bilayer of phospholipids. Tautology, sorry about that. And in the phospholipid bilayer, we are going to have multiple different important proteins, and there are 3 important proteins in the envelope of the influenza A virus, and these are the hemagglutinin protein, the neuraminidase protein, and the M2 ion channel. So now let's start by actually putting some of these proteins on. So we'll start by putting on hemagglutinin, and we'll have this in red here. So, the envelope of the virus is going to contain lots of these hemagglutinin proteins, and I'll go over the structure of these later on. But for now, we're just going to represent them as this little spike in the envelope, and you won't just have one of these, you'll have multiple of these. So, where should I put its label? I'll try and squeeze it in here. So, for short, this is abbreviated down to HA. Its full name is hemagglutinin, so this is the hemagglutinin protein of the influenza A virus here. And you won't just have one of these molecules in the viral envelope, you'll have loads of them. I'm just going to draw one of them to keep it simple, but in reality you'll have multiple of them in the viral envelope. The viral envelope will be studded with them all over. So this makes the virus look spiky actually, because it's got loads of these hemagglutinin proteins sticking out from its envelope out into the outer world. Now, we will come to this later on, but the hemagglutinin protein is really important for binding to host cells so that the virus can then enter those host cells. So this is the initial thing that grabs onto molecules on the surface of host cells. So that's hemagglutinin. Next important protein is neuraminidase. So we'll do this in yellow. Now, neuraminidase, overall, to draw a basic shape for it, it has this sort of mushroom shape, which I'll try and do here. So this is representing a neuraminidase protein in the viral envelope. So, for short, we can abbreviate neuraminidase to NA, and indeed I will be doing for the rest of the video, but in full, the name of this is neuraminidase. Now, sometimes you will see the neuraminidase protein called something else. You will also sometimes hear it called the sialidase enzyme. And you will understand these two names later on when we discuss what it is that hemagglutinin actually binds to. Neuraminidase for now, or sialidase you can also call it, for now just take these as names, and the function of neuraminidase, just to paint a picture of where we will go, and I will explain this much better later on, is when the virus particles are budding off from the host cell. So hemagglutinin is important for the virus just entering a new fresh host cell. When the new viruses are coming out from the host cell, and remember, viruses are intracellular parasites. They go inside the cell and they use that cell to make loads of new viruses. So hemagglutinin is the particle that the virus uses to get into the cell. Neuraminidase is important for the virus particles being able to get out of the cell. So when you've created loads of new virus particles, neuraminidase is really important in the budding off process. But we'll see all of this much later on. And again, I've drawn one neuraminidase there. In reality, you'll have loads of them. Not quite as many as you have of hemagglutinin. I think the ratio is usually quoted as you have 4 times the amount of hemagglutinin as compared to neuraminidase, but you still do have lots of neuraminidase proteins in the envelope of the influenza A virus. Okay, then the next protein, the final protein that is in the viral envelope of influenza A viruses, is the M2 ion channel. Whoops, sorry about that. And this, as the name suggests, is an ion channel, so I'll draw it like an ion channel. So here in blue, like so, and there is the pore down the middle. And this is an ion channel that conducts protons, and it is going to be extremely important in the entry mechanism for the virus again. Its role comes after hemagglutinin, but it's also very important in the virus getting into the host cell initially. So this is representing what is called the M2 ion channel. Now, the influenza B viruses and the influenza C viruses are also going to have an M2 ion channel, and these M2 ion channels of the different species of influenza virus have slightly different properties. Therefore, sometimes, to stress that we are talking about the M2 ion channel of the influenza A virus, we put an A in front of it. So you might see this called AM2. All that means is the influenza A virus M2 ion channel. If you see BM2, that means the influenza B M2 ion channel. If you see CM2, that means the influenza C M2 ion channel. That will make more sense later on, but you can just call this the M2 ion channel at this stage. Right, and again you'll have multiple M2 ion channels in the viral envelope. I'm lazy and I'm only drawing one of those. Right, and it's a proton channel, remember, that's the ion that it conducts. So those are the 3 proteins that are going to be in the viral envelope. Let's now go inwards. So what's underneath the viral envelope? Now remember, membrane, phospholipid bilayer, is gloop. It doesn't have any sort of structure on its own. This thing would be blobbing around, changing structure continuously. Imagine that, it oscillating, it becoming thinner and then getting thick again, it would be like jelly. It's not structurally sound at all. The virus would not stay in a perfect sphere all the time if it was just like this. So underneath the phospholipid bilayer, you have a structure that actually is strong, that holds the phospholipid bilayer up and keeps the virus in a perfect sphere. And this is made up, it's a spherical shell made up of loads of proteins called the M1 proteins. So let me now draw these in, and I'll do it in green. So here we go. Underneath the phospholipid bilayer, you have loads of these proteins. So each one of these blobs is a protein, and they form a great big spherical shell. So loads of them are bound together. They're little globular proteins, and they form a great big shell, like so, that goes all the way underneath the phospholipid bilayer, like that. And the key thing that I've tried to show here is, look, when we have M2 ion channels, they penetrate this M1 channel, like so, so that if this was to allow protons through it, it will conduct them into the inside of the M1 shell. And that is important, that the protons end up coming in to the inside of the M1 shell here. So all of these proteins in green here, these are M1 proteins. And they have another name as well that's often used. They're often also called matrix proteins. And that's because this shell is referred to as the matrix of the influenza A virus. Now, an important point that people often misunderstand here is that people think that this M1 shell that we formed here, that is just underneath the phospholipid bilayer, it's spherical and it holds the phospholipid bilayer in this sphere, they think that this is the capsid of the influenza A virus. It is not the capsid of the influenza A virus. We refer to this as the matrix of the influenza A virus. We're going to see that inside here we are going to have the genome of the influenza A virus, and the genome is not going to be just a single piece of RNA. In fact, it's going to be a segmented genome. We're going to have 8 separate pieces of mRNA that are coding for different proteins, and each one of those is going to have its own capsid. So we do not refer to this as the capsid of the virus, even though looking at this picture you might be tempted to think that this is the capsid of the virus. It's not the capsid of the virus. Okay, so underneath the envelope then we have this M1 shell that holds the membrane up. And overall, this shell, even though my picture does not suggest it, consists of around 5,000 M1 proteins. So I might just actually write that down here. So you put 5,000 or thereabout M1 proteins together to make this beautiful spherical shell, and then the phospholipid bilayer, the viral envelope, sits nicely on top of that, and it's held in this spherical shape by the M1 protein shell. And also an important point is that this binds to these proteins in the viral envelope. It binds to their tails down here of the hemagglutinin and the neuraminidase, and also the M2 ion channel. And the other key point that I told you about is the fact that the M2 ion channel penetrates the M1 shell, so that if this was to allow protons to come through, they will be coming into the inside of the M1 shell. Okay, right, so that's M1 protein done. Let's now go inside further. Let's go now to what is inside of the M1 shell. And inside here we're going to now have the viral genome. However, I just want to talk about one more protein before we come on to the viral genome. So inside here we are going to have the viral genome, and I'll add that on in just a moment, but there is also, along with the viral genome, another protein which we didn't think initially was inside here and was given a name that suggested that it was not inside here. So this is a virally encoded protein, so it's coded for by the viral genome, and I'll just put it in here in this pinky sort of colour here, and this is called the NS2 protein, or at least that was its old name, and it's a name that's still often used. It's now got a new name. It's now called the NEP protein. So what do these stand for? NS2 used to stand for non-structural protein 2. So when we first found this protein, we realised that it was a viral protein. We realised that the viral genome of the influenza A virus coded for this protein, but we did not think it was actually a protein that was used to make virus particles. We did not think the virus particles contained this protein. We thought that, like the non-structural protein 1, which we'll talk about later, which isn't part of the virus, which truly is non-structural, that it had some role in the viral life cycle, but it wasn't an actual structural protein. And then it turned out that actually inside the M1 shell of the influenza A viruses you do have NS2 proteins, and therefore we have come up with a new name for new non-structural protein 2. It's now the NEP protein, and this is a good name for it because it describes actually one of the major functions of this protein, which is nuclear export. So this is the nuclear export protein. So what does this protein do? It exports fresh, new viral genomes that have been created in the host cell that has been infected by the virus. It exports them out of the nucleus, and then it takes them to where the new virus will form, and this is why it ends up actually being inside the virus particles, because it was in the right place at the right time, basically. So it's involved in bringing new viral genomes that have been created out to the surface membrane of the host cell where the new virus particles will be forming, and therefore it ends up inside the virus particles as well. So it is a structural protein, in fact, hence why we needed to rename it from NS2. However, it hasn't really caught on as well as the people who decided to rename it would have hoped, and people do still call this protein NS2. In fact, most people now call it NS2/NEP, so I might just put this here. So it's often called NS2/NEP. Excuse me. Okay, so that's another viral protein then. Let's now go on to the interesting stuff, the viral genome. So as I say, the viral genome of influenza virus, there are lots of adjectives that we can use to describe it, and I'll use a few of these now. So let me firstly decide on a colour. Let's use purple. So it is an RNA genome, and it is a single-stranded RNA genome. So the nucleic acids that will be encoding the genetic information are going to be single-stranded molecules of ribonucleic acid rather than DNA. And these pieces of RNA are going to be negative, and that's just a shorthand for negative sense RNA. And what that means, as I described earlier, is that you cannot actually use these pieces of RNA to translate into protein. If you attempted to translate them, you would end up with a horrible mess of a protein. You would not end up with the proteins you wanted. Instead, you need to take the complementary piece of RNA, you need to take all the complementary organic bases, and then that is a piece of mRNA that you can actually use to translate and produce proteins. That is what we would call a positive sense piece of RNA, a piece of RNA that can actually be used, can actually be translated to produce the proteins. Influenza is not gifted, unfortunately, with positive sense RNA, which makes it, gives it a bit more work to do in order to actually make proteins. It has negative sense single-stranded RNA genome. The other key adjective to add in is that its genome is segmented. And this means that it's not a single piece of single-stranded RNA. In fact, you have multiple different pieces of single-stranded RNA that code for the genes, just like in humans. I mean, we have 46 chromosomes, 23 pairs of chromosomes. All of our genetic information is not on one great big piece of DNA. And it's the same with the influenza virus. They have multiple different pieces of RNA coding for their genetic information. Now, you might be expecting me now just to actually draw on these pieces of RNA, just draw, I don't know, and here's another key piece of information. In the influenza A virus, how many pieces of RNA do you have? You have 8 pieces of RNA, so not quite 46 like in human cells, 8 is the number in influenza A virus. So you might be expecting me now just to draw on 8 pieces of single-stranded RNA onto my picture here of the influenza A virus. I'm not about to, because these pieces of single-stranded RNA are not alone inside here, and I said this earlier on. They all have their own little protective capsids, which are made out of proteins. They also have, accompanying them, an enzyme complex that is an RNA-dependent RNA polymerase. So, these pieces of RNA are negative sense. That actually is a problem for the influenza virus. That is not good. It makes its life much more difficult, because if this is going to come into a human cell and wants to produce its proteins, it needs to somehow make from its pieces of RNA complementary RNA, and then it can use those as messenger RNA to be translated into proteins. That is difficult because human cells have absolutely no need for an enzyme that can use a piece of RNA and synthesize a complementary piece of RNA. Remember, in human cells, we make RNA from DNA. We have enzymes that go along DNA and make a complementary piece of RNA, so-called DNA-dependent RNA polymerases. We do not have enzymes which can go along a piece of RNA and synthesize a complementary piece of RNA, which are called the RNA-dependent RNA polymerases. Therefore, the influenza A virus is going to have to bring in its own enzyme to do that, and indeed each segment of the RNA genome here is going to have its own enzyme for doing that. So actually, each of these pieces of RNA is in a complex with loads of proteins, and these complexes are referred to as ribonucleoproteins. So let me get another colour because I really don't actually like this boring purple. Let's go for a more vivid purple. Okay, so all of the RNA pieces are in these complexes called RNPs for short, and this stands for ribonucleoprotein. So the R is for ribo, and then the N is for nucleo, and then the P is for protein. So RNP for short, and of course we're talking about multiple of them, so ribonucleoproteins, RNPs. So what does this mean? Well, the term nucleoprotein refers to when you have a complex involving a nucleic acid and a protein. So when you've got some sort of nucleic acid molecule bound to a protein. Ribonucleoprotein, therefore, it's too easy, means when you've specifically got a ribonucleic acid, when you've specifically got some RNA complexed with protein. So a ribonucleoprotein is literally just a piece of RNA bound to a bunch of proteins, or multiple pieces of RNA bound to a bunch of proteins. It's a complex that contains RNA and proteins. In reality, the ribonucleoproteins of the influenza A viruses are just going to contain a single piece of RNA with lots of proteins. So, let me add these onto the picture then. So I need to somehow try and fit in 8 of these. Whoops, pen. So I'm going to try, so I'm just going to represent these then as purple lines, otherwise it's not going to fit in. So here we go. 1, 2, 3, 4, and then I'll have to be cheeky and put one in like that. So 5, 6, 7, 8. Okay, so there we go. Each of those purple lines is representing not a single piece of RNA, even though it looks as though it is. It's representing the RNA complex with a whole bunch of proteins. It's representing the ribonucleoproteins. Now, let's take one of these pieces out and actually have a look at its structure in more detail. So, I'm just going to move a little bit, like so. So, of course, one of the components of the ribonucleoprotein, in fact the only component we know at the moment for sure, is the piece of RNA, and each one of the 8 ribonucleoproteins is going to have a different piece of RNA. So I'll start by drawing the RNA here. So in purple, that's representing the RNA. Now, I said that each of these pieces of RNA is going to have its own capsid, its own bunch of proteins to protect it. And this capsid is going to be made out of a protein that is called nucleoprotein, or NP for short. So, the capsid protecting each piece of RNA in the ribonucleoprotein is just called nucleoprotein protein, or NP for short. So let me add these onto the picture. So you don't just have one of these, you have loads of these, and they bind all the way along the piece of RNA. So like so. In fact, I'll put one there as well. So these in yellow, these are representing nucleoproteins. These blobs, each one of the blobs is a nucleoprotein. So like so. So all the way along the RNA molecule, you have these nucleoproteins binding to the RNA molecule and protecting it. So here is the start of the ribonucleoprotein. In fact, you could refer to this as a ribonucleoprotein already because it's a complex between protein molecules and a piece of RNA. Now, it's made more complex. You do not just have it like this inside the influenza A virus. It would be beautifully simple if it was like this. Unfortunately, it twists into a bizarre shape. So let me try and show you this bizarre shape. And I just want to go over the piece of RNA to make it more obvious. So here in purple, here's the piece of RNA again. Now what I'm going to do is actually extend the piece of RNA, so I'm going to be cheeky. I'm going to say, actually, the piece of RNA was this long rather than this long. Okay, so now this piece of purple is representing the piece of RNA, and again, all of the length of the RNA will be bound to nuclear proteins, so I'll have to draw in a few more nuclear proteins. So all I've done is modify the picture there. So here now, this is exactly what I had before. I've just bent it round, we're imagining now. So I've got my great big piece of RNA, and it's still complex with these nuclear proteins. So you might say, well, that's not too complicated. I can understand a piece of RNA being bent round like that. But the more complicated bit is now what you need to imagine doing is twisting the whole thing. So imagine holding this here. So you hold those two bits in fixed position, and then you start twisting this thing around so that you get twists in this, and that is the way that these ribonuclear proteins are going to exist. So they're the RNA twisted around with these nuclear proteins bound to it, and I am not going to attempt to actually draw that, I'm afraid. I'm hoping that your imagination can come up with a picture of what this would look like. Alternatively, you could go onto Google and type in influenza A virus ribonuclear protein, and you'll get a beautiful picture of this. Okay, but that's what these ribonuclear proteins are, effectively. They're the piece of RNA with loads of nuclear proteins bound to it, and they form the capsid. This is now referred to as the capsid of the piece of genome, and then the whole thing twists around, so it's twisted all up like that. Okay, the final thing to complete the ribonuclear protein is we need to have the attached RNA-dependent RNA polymerase, and now I have to write that ridiculous line out. So let me choose a colour, we'll go back to green. So, along with the RNA and the many copies of nuclear protein, you are also going to have this special enzyme which is the RNA dependent, meaning that it uses RNA, and then it's an RNA polymerase, which means that it's going to synthesize a piece of RNA. So it synthesizes a complementary piece of RNA from a piece of RNA. And I'll repeat, human cells do not have these. They do not have need for these. They have DNA-dependent RNA polymerases, which synthesize a piece of RNA from DNA, not from RNA. And these enzymes are different. A DNA-dependent RNA polymerase, I'm afraid, cannot synthesize a piece of RNA from a piece of RNA. Now, for short, I will often abbreviate RNA polymerase down to RNAP, RNAP for short. Okay, so RNA-dependent RNAP then. So this is going to consist of 3 separate proteins, this RNA-dependent RNA polymerase, and these proteins are encoded for by the viral genome itself. So let me draw these on here. So here then, I'm just deciding how I want to draw this. So I'll give it its classical sort of enzyme shape. So here we go. Whoa, damn. There we go. That's fixed that up. So, this is the RNA-dependent RNA polymerase. Now, unfortunately, this is not made out of a single polypeptide. There are 3 polypeptides joined together to make a protein complex that is the enzyme. So I need to now split this into 3 separate components, which I'll just do in a very basic way, like so. So that is meant to represent the fact that this is built out of 3 separate polypeptides that are all coded for by the viral genome. So in these 8 ribonuclear proteins of the influenza A virus is the information for making each one of these proteins, and these proteins are given rather boring names. One is called the PA protein, the next one is called the PB1 protein, which can be the one in the middle there, and then the final one is called the PB2 protein. So these are 3 proteins, PA protein, PB1 protein, PB2 protein. I'm not aware of what they actually stand for. I don't know if they stand for anything. They probably do, but it's not important. And these 3 proteins join together to make this RNA-dependent RNA polymerase, and each one of the ribonuclear proteins has one of these RNA-dependent RNA polymerases. So this thing that I have kind of drawn here, if you imagine this twisted up, that is then the picture of a influenza A virus ribonuclear protein. So for each one of these purple lines, this is what you got. A piece of mRNA with loads of copies of this nuclear protein bound to it, and then the RNA-dependent RNA polymerase, which consists of these 3 separate proteins. Okay, so you'll be relieved to know now that that picture that I have drawn you here, that is it. That is an influenza A virus. That is everything. Now, you will have more than one non-structural protein too. You'll have multiple of those, so I'll just add another one in there. In fact, actually, I don't know why I've done that, because I didn't add multiple M2 ion channels, hemagglutinins and neuraminidases, so I've kind of been a bit unfair there. But all of these proteins, the M2 ion channel, the hemagglutinin, the neuraminidase, and the non-structural protein 2, you have more than just one. Okay, so next piece of information I want to give you is about the viral genome segment. So I want to make this a little bit more clear. So we know we have 8 pieces of single-stranded negative sense RNA that make up the viral genome, and they're beautifully protected in these ribonuclear proteins that I've now spent so long describing to you. These viral genome segments are given names, and they're given the beautiful names 1, 2, 3, 4, 5, 6, 7, 8. Now, the next piece of information then that I want to give you is which proteins, which influenza A virus proteins are coded for on each of the viral genome segments. So let's do this. So we'll start with number 1 then. So on genome segment number 1, there is a single gene, and this is the gene that codes for the protein PB2, which of course we know exactly what that is now. That is a component of the RNA-dependent RNA polymerase that is associated with all of the ribonuclear proteins. So on viral genome segment number 1 is the PB2 gene. On viral genome segment number 2 is the gene for PB1. Next, on viral genome segment number 3, again there is only one gene, and it is the gene for the PA protein. So this is all going rather well. We're seeing loads of proteins that we know the names of that are coded on these different viral genome segments. So this is all making lots of sense. Let's continue on, and it continues to make sense. So on protein number 4, on genome segment number 4 is the gene for the hemagglutinin protein, so HA. So only one gene again. On genome segment number 5 is the gene for the nucleoprotein, so NP. On genome segment number 6 is the protein for neuraminidase, NA. On genome segment number 7, it then gets a little bit more complicated. We have more than one gene on genome segment number 7. We have the genome segment for the M1 protein, and we also have the gene for the M2 protein. So M1 and M2 are both on the same genome segment. They are both on genome segment number 7. Finally, on genome segment number 8, of course, there's going to have to be the non-structural protein too, because we haven't seen that on any of the other genome segments, but there is one other protein that I've talked about but we haven't seen yet, which is the non-structural protein 1. So, that is the final protein of influenza virus A, non-structural protein 1, NS1, and then there's NS2 also here, which is also called NEP. So there you go, those are the genes that are on each of the 8 viral genome segments. So overall we have seen that influenza A viruses have these 8 genome segments, and overall how many proteins are coded for on these 8 genome segments. Overall, it's 10 proteins. 9 of them are actually on my picture of the virus here, and I'll just count them so that you believe me here. 1, 2, 3, 4, 5, and then there are 4 in the ribonuclear proteins. 6, 7, 8, and then the nucleoprotein, which makes 9. And then there is one that is truly a non-structural protein and is not in the virus particles, and that is the NS1 protein. We will talk about the NS1 protein later on. It's involved in stopping host antiviral responses, but we'll discuss that in much more detail later on. Okay, so there you go. We have now discussed all of the different proteins that are coded for on the different genome segments of the influenza A virus. I think we'll take a break here because we've been going for 40 minutes. In the next video, we will continue on discussing the influenza A virus. I want to go into more detail about the hemagglutinin protein. I want to go into more detail about the neuraminidase protein. I want to go into more detail about the M2 ion channel. Then I want to discuss a little bit about all the different strains of influenza viruses. I want to discuss what it means when people talk about a strain of influenza A virus being H1N1. What does that actually mean? So we'll handle that in the next video.